cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of 3 bases in the nucleus. The DNA code determines the order in which the 3 bases are added to the messenger RNA. This process is called transcription. Before a messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, I'm going to play that one more time and then we'll kind of talk about it as it's going.
Here is a cell, the basic
Now,
unit of all
can you
living
look at
tissue.
this cell? Do you recognize some of the structures that we learned in the last chapter? You see this?
The nucleus contains the genome.
Endoplasmic
In
reticulum.
humans, the genome is split
The
between
nucleus has
23
little
pairs
pores
of
in it.
chromosomes.
The chromosomes,
Each chromosome
there's
contains a long strand
23 of
of
those.
DNA,
Have you ever heard of
tightly
23
packaged
and
around
me?
proteins
You
called
can get your
histones.
DNA tested. That's why it's where the 23 comes from.
Within the
The
DNA
histone
are
is just
sections
to
called
make the
genes.
chromosomes
These
stronger,
genes contain the
not so
instructions for
fragile.
making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of 3 bases in the nucleus.
And look at the letters here. We have
The DNA
A,
code
T,
determines
C,
the
and
order
G
in
are
which
the
the 3
three
bases are added
or
to
4
the
kinds
messenger
that the
RNA.
DNA has. But notice what
This
matches
process
with it. There's
is
no
called
T in there.
transcription.
Instead of a T, there's a U. But each one is always matching up with its partner.
Before a messenger RNA can be used as a template for the production
Okay,
of
now
protein,
this is the
it needs to be processed.
initial messenger
This
RNA,
involves
but
removing
not
and
everything
adding
is
sections of
useful.
RNA.
So it's going to snip out the pieces that it needs and
The
put them back
messenger
together.
RNA then moves out of the nucleus
So we'll use words
into
like
the cytoplasm.
intron and exons. Okay, now it leaves out through the pore, out to the
Protein
cytoplasm.
factories in the cytoplasm called
Okay,
ribosomes.
and the ribosome's going
bind
to
to
come
the
along and
messenger
attach
RNA.
to it and start
The
reading the
ribosome
messenger
reads the code in the messenger
RNA
RNA
instructions.
to produce a chain made up of amino acids.
Okay, this is the transfer
There are 20
RNA,
different types of amino acids.
and these are the
Transfer RNA molecules
three-
carry the amino acids to the ribosome.
these are the anticodons. These
The
are the
messenger RNA
anticodons.
is read 3
And
bases
these are
at
the
a time.
codons. So notice that this thing
As
going
each
too
triplet
fast
is
from
read,
here.
a
But
transfer
every
RNA
3 of those
delivers
is
the
the
corresponding
bottom part's the
amino acid.
codon. The matching part that goes with that is the
This is added
anticodon.
to a growing chain of amino acids. last amino acid has been added, the chain folds into a complex 3D shape to
And you
form
might ask the
a
question,
protein.
how does it know when to start and how does it know how to end? We'll talk about that. And then when it's all done, you might ask the question is, why does it fold? And this is something that I've only learned, and I don't know how long we've actually known about this. I usually learn these things after they figured it out, but that different amino acids may have different charges on one part than on another, and like you know how that opposite charges attract and like charges repel, and if there's more than one charge on there, you might have doing a mixture of things. So like if this amino acid and this amino acids are oppositely charged, then they attract to each other, so it'll fold at that spot. And then the next one, that's attached onto here, well, how is its charge compared to this? If it's opposite, it might be straight at that point. And if it has a charge that's off on one side and is now near this one down here, then it'll fold down this way. So you can see this thing is folding and just getting all kinds of different shapes as they wrap around each other. But initially it probably starts out fairly straight and then starts folding on itself until finally it has its particular shape. So every protein kind of ends up with its own unique shape. But it's because what one amino acid is doing to all its neighbors. Once it's folded, its neighbor might not be the one only next to it. It might be one that's down a ways, but now it's next to it, and so that may call it the bend, one way or the other way. So the stuff that I'm telling you here, I never learned when I was in high school because they didn't know about it. Didn't learn in college, they still didn't know about it. A lot of this stuff it's come about since I've been teaching. So let's go to your note packet, and we're going to we'll kind of go faster on this part. Yep, good. Working. So an RNA, the messenger RNA that it made there, notice it was just a single strand. It just had the little thing sticking off one side, is the way it showed in the animation. It was just like half of a ladder, right? So that's RNA. One thing that there were always patterns that it had. Take away a little bit of chemistry stuff here. So the pattern is on the DNA, you always had adenine pairing with thymine, and you had cytosine always pairing with guanine. So the letters we used were A, T, C, and G. Okay? This was for DNA. RNA, the messenger RNA, when transcription took place, if there were instead of a T, it uses a U instead, but it still is a C and a G. These sorts of patterns. So let's say we have a messenger RNA, and we have, let's just make up some letters here. Let's see, A, U, C, U, C, C, let's see. Oh, we got to have a G in there. Let's put a G here. Okay? So every three of these is a code. So this is the messenger RNA right here. We're going to call it mRNA for messenger RNA. And every three of those is a code, the codon. Then when the transfer RNA with its amino acid comes along, the transfer RNA has a code of its own. but it's called the anticodon. Why do we call it anticodon? It's kind of always like the opposites. So the opposite of an A is a what? A U. So in the transfer RNA, the transfer RNA needs to have this code for it to match up to bring the right amino acid. So with an A goes U, with the U goes A, and with the C goes G. So so the bottom part here is the codon. This is the anticodon. Remember the structure that looked like this? Looked like kind of like what do we call those? Gingerbread man. It had the shape like this, if you're a gingerbread cookie, and its legs are sticking up, and on the end of one of its legs is where the amino acid is attached. If you kind of want to have something funny in your mind to kind of picture it, it's like an upside down gingerbread man. On one of its legs, the amino acid attaches. On its head, he has three pointy things, three horns sticking out, and those are the anticodons. And the amino acid, the transfer RNA, this is the transfer RNA. When it attaches on here, it will only attach where it has the right codon to match with it. If it doesn't have the right codon to match with it, it can't bond on. And it won't, so we don't get the wrong amino acid being attached and making the wrong protein. Now, do you remember she talked just very briefly about this piece that was taken out of the messenger RNA? So you have this pre-messenger RNA, pre-mRNA, and then it took out some pieces. The pieces that it takes out are the introns, and the pieces that are left behind are the exons. And so it's sort of like it edits the message to only include the pieces that are absolutely necessary to make the protein. So if this isn't necessary for the protein, it edits that off. If this isn't necessary here, it edits that off. If there's pieces inside of it doesn't need, it takes those out and just puts together kind of like a condensed version of everything you need to know to make that protein. Now, it does have two things in the front and the back. It needs to have one code that says start, another code that says stop. Okay? So every one of those messengers, RNAs, for making a protein has a start, then the codes for the amino acids, and then a stop. So if you think about this whole process that we've been talking about, there's three kinds of RNA. There's the one that carries the message from the DNA that we formed by transcription. That's where the ladder unzipped itself and it made the other half. And then when this moves off, this thing will zip back together again. Then we have transfer RNA. Their job is to attach to the appropriate amino acid. And so that, in other words, this transfer RNA doesn't match up with every amino acid. It only matches up with one. So each amino acid has to have its own transfer RNA. And then there's the ribosomal RNA, whose job is to take the message using the raw materials from the transfer RNA brings in, and to assemble the protein, assemble the amino acids to make a protein. So here's that, where I was thinking about. Here's that original message, let's say, and these are always codes, but some of them aren't needed. So it will use an enzyme to take out the pieces that it doesn't need and then reassemble it. Now, here's the part that I find very fascinating about this. You think about how complicated this process is of just making one protein. Everything has to work perfectly. And how well does that go? Well, look at yourself. you're pretty much working, I mean, you work fine, right? Every part of you, you don't have, something sticking out of here or here, it looks like, you look like almost every other human being, right? Other than the surface features, they're a little bit different. So this process works fantastic. Now, can it fail? Yes. We live in a fallen world, and yes, it does fail, And those are different diseases, that people can have because of that. Okay, so we take the initial message, then we edit it, and this is the piece that goes out of the DNA, out of the nucleus, and goes out into the cytoplasm where the protein's going to be made. So this is the one we want to use. We don't want to use this one, because this contains information we don't need. This only contains the pieces that we need to make a protein. The transfer RNA, remember that's the gingerbread man that's standing on its head with the 3 horns sticking out the bottom I was talking about. These horns are the anticodon. So it's the anticodon horn. Or would you rather have like a crown? I don't know, whatever you want to call it. But these are, this combination right here matches with whatever amino acids at the top. They have to go together, otherwise we're making proteins that your body may not need. Or maybe proteins that would actually harm you. So we don't want that. We want to make the right protein. So they transfer amino acids from the cytoplasm to where the ribosomes are, because the protein is assembled by the ribosome. The protein is assembled by the ribosome. It takes the message from the messenger RNA using the amino acids brought by the transfer RNA to make the protein. So here, this picture that you have in your book is maybe not as puffy and convoluted as the one that was in the animation, but they're both brown. That's the only thing they have in common. But this is the thing that's responsible for making all of it happen. Okay? Here's the message that came from the DNA. Here's my transfer RNA coming in. Why is it going to be able to attach there? Because this anticodon exactly matches that code. There's probably lots of other transfer RNAs out there, but they don't match. But only the ones that matches, and then the ribosome then attaches this onto the end of the other amino acids that are already there, or that already have been attached. And we're making, so this long chain of amino acids is sometimes referred to, or usually I should say, not sometimes, usually referred to as a polypeptide chain. Polypeptide chain. So that process of assembling them all together to make a protein is called translation. You kind of think of it as translating the message into a real protein. The message was on the messenger RNA, and now we made a protein out of those instructions that were on the messenger RNA. Remember it said that the messenger RNA leaves the nucleus through that nuclear pore. The pore are the little holes in the nucleus that only let certain kind of things out and in. A ribosome lines up at one end of the messenger RNA. So in other words, it always starts at one end, and it's going to start at the end where it has a code for start. So that's how it knows which end. It's looking for the codon for start. And almost every living thing uses only a few codes for start. There's a lot of living things where the code for start is the same as in the next kind of living thing. So in other words, the starts are pretty much universal. And that starts at that end, starts assembling it. When it gets to the other end, there's going to be a code that says, stop, no more. Okay, that's the end of the protein. So one signals the beginning of making a protein, the other one signals the end. I'll skip over these a little bit too fast. So the amino acids kind of hop off that transfer RNA. There's a process that it goes by, but we won't worry about how it hops off. And how it goes about actually getting bonded on to the other to make that polypeptide chain. So once the amino acid bonds to the chain, that transfer RNA has done its job, and it's going to be used again to go out and find another amino acid of the very same kind. So as long as those amino acids are out there, their transfer RNA is going to find it. I don't know, we haven't been instructed in the book in any way. How does it know about going to find it? Is it just pure chance, or is there something that tracks them? Would be my guess. Maybe God put something in there so that it kind of knows what to look for. Okay, here's where I was talking about. There's a start codon. In other words, it's 3 letters, and these 3 letters only code for start. If you look on page 92 in your textbook, you notice that there are some of them that say start, I think it is. Well, here's one that says stop. On the start, the start is down there towards the bottom left where it says start. And it's, so there's several stops, but there only shows like one start, for instance. But there are a couple other starts. But so it's not like there's as many different kind of starts. It does just a couple, So if that codon was in some other living thing, it would mean the same thing. Now here's a little extra detail. That messenger RNA that contains the blueprint for that protein, there could be a couple of them working on that same message. One starts it, and then as it's farther on down the line, another one can start making another protein. So we can have more than one ribosome working on the same set of instructions. They're not at the same point, obviously, but they're both trying to make the same protein. So the messenger RNA can be, and also the messenger RNA can be used more than once. So it doesn't make one protein and then have to get the message all over again. It can use that same message again and again and again. Okay, so that finishes up our discussion about protein synthesis. Next, we have the topic of metabolic rate. That's the rate at which organisms carry on all these life processes that we talked about, that living things have to be able to do. Metabolic rate is, that's what determines all the living processes that help you to maintain that steady state. That homeostasis, remember, means steady state. You're trying to maintain a steady state. And so there are certain things that have to happen for you to maintain that. There are certain things in your body, for instance, that need to be replaced, repaired, things like that, so that you can continue to live or to exist. In the topic of metabolism, we have 2 words. One's called anabolism, and the other one is catabolism. This first word, you may have heard that not with an ISM on it, but with an IC on the end. Anabolic. You ever heard what the next word is? Bodybuilders use it. Not all of them, but some of them do. We want to have the big, massive bodies. What do they call those? They use what? Steroids, right? The full name is Anabolic Steroids. So just think about that and why they use it. And then there's another one called catabolism. So there's anabolism and catabolism. Anabolism is a process where you're building up molecules and storing energy. So now does that make a little more sense why why we call it anabolic steroids. We're giving them a steroid to stimulate the body to build up more molecules. More muscle mass is usually what they're after. Yes?
If you, like, some athletes like die earlier though because of like taking too much steroids or something, right? Because their body can't.
Yeah, steroids are really, well, if used properly, you know, like when doctors use it, steroids are typically used when the body is doing something and it doesn't know how to quit. You know, like for instance, sometimes, and it's interfering with healing or something like that. You know, like sometimes if some people will have to get some steroid shots to maybe like to stop a pain cycle. You know, they're just, and because of the pain, you know, it just kind of, it's a vicious cycle like that. So basically, it's stopping the body from responding in a certain way for a period of time so that things can kind of go back to normal. And then we wean them off of that steroid so that hopefully that whatever caused them to start this process is gone and things can go back to normal. My wife had an autoimmune disease. It's probably now, I don't know, like 30 years ago. It was called vasculitis. It's an infection of the blood vessels. And it was one of those where she had to be on prednisone. You've probably heard of that before, prednisone, some of you. But is to stop her body from going through this cycle of inflaming her blood vessels. And it's a very slow healing process. It took almost a year for her to get over it. But hopefully at the end, you know, that the body is now finally back into its normal routine, and then I wean off of it and life can go back to normal. And for the most part, it has. She's had a couple incidents that we wonder if they were, you know, as a result of that original autoimmune disease that she had. The downside also is, like you've talked about, of taking steroids or, you know, like that, that's really not, it's really not good for the body, but whatever was happening was even worse. And so we're trying to stop something, but we want to get off it as quickly as possible. One of the things that they were concerned about my wife being on for almost a year, because usually people aren't on for anywhere near that long, is that your bone density diminishes, and so if you were to fall, you know, your bones would be more likely to break and things like that. And thankfully, God didn't have that as one of her side effects, but you sure could have that. So anabolism is a process of building up. Catabolism is a process of breaking down molecules. Now, let me give you an example that we will talk later on when we talk about amphibians and particularly frogs. Frogs start out as what? As tadpoles, right? Do they have legs? Nope, not originally, but they have a tail, right? Okay, tail. Well, as they mature, they have to have a catabolic process taking place. They need to have the tail be absorbed. In other words, torn down and got rid of. And it will use those raw materials in a process called anabolism, to build the molecules necessary to have front feet and back legs, right? So there's an example of something where you have to have both. Metamorphosis in a caterpillar to a butterfly. They have the catabolic process. The part that's almost amazing is that if you were to look inside of one of those cocoons, it's almost like, it's just almost pure liquid inside of it. Where's the butterfly? You know? But the instructions are still all there to make a butterfly. But it tears everything down and then reassembles it all into something with wings and legs, you know, only, you know, like 6 legs as opposed to all the little legs it had before, you know. So, but the instructions for both were there. One gets turned off. We're not trying to make another caterpillar. We're trying to make a butterfly. And so it uses that set of instructions that have to do with making wings and stuff that we normally associate with the butterfly, with the little proboscis, you know, for sucking up nectar and stuff like that. So here is an anabolic process that maybe a cell would do. It would absorb food like we talked about before, and then down here we have a lysosome joining it to dump in its enzymes to digest it. The raw materials are absorbed out into the cytoplasm, and the parts it doesn't need are dumped outside the cell. But it's going to use these molecules there to build, or materials to build the molecules it needs for the cell, you know, the parts that it needs for the cell. So this would be an example of what we call intracellular digestion. Food is taken inside the cell and then digested. Another kind is extracellular digestion. That can also happen. How long will it be? Nobody knows. Have you ever Venus flytrap? Okay, you know, fly gets in there, triggers it, and it closes up. But does it chew the fly? No. It secretes enzymes outside its cell, digests the fly outside of the tissue, and then absorbs it. So that would be an example of like extracellular digestion.
So how fast does it close?
But the little hairs on the side kind of help it, but it doesn't go real fast because I think if it did it, it'd have to be really, there's kind of like a border between going too slow and too fast. If it went maybe responded too fast, the fly might, you know, quickly respond. But I think it goes just slow enough that these are already crossing over by the time the fly realizes, I need to get out of here, and now it's too late. I've watched them close before. I mean, you can, every once in a while, you find places that are selling Venus flytraps, and you can buy one. They're not all that expensive. But they kind of go, you know, like this, and then the last part's a lot slower, you know, because there's no chance the fly's going to get away anymore. The holes are too small. Now, autophagy. Auto means what again? Self, right? Does it mean self? And in phagy, and we had phagocytosis, that meant what? Feeding. So this is self-feeding, where a cell feeds on parts of itself. Well, that sounds like a bad thing. But what's happening here is there might be some structures inside the cell that are damaged maybe in some way. And so, remember I said that God made living things as masters of recycling? It will take that structure using catabolism, that process, and tear it down into the raw materials, probably dispose of the things that are defective, and add some new parts in there, and then rebuild the structure. So it doesn't have to have a new one every time. It uses as many of the materials that are still usable and rebuilds it again. Yes, it needs some replacement materials, but it will make this new structure. So this is a time when it's going to catabolize a defective structure and then anabolize a new structure.